Dr. Nathan got his Bachelor of the Arts from Harvard College and then went on to Harvard Medical School to obtain his MD. And from there he took a faculty position after some training, of course, at Rockefeller University, where he rose to the rank of associate professor before he was recruited away by Cornell University Medical College, where he is now a professor of microbiology and immunology and a professor of medicine, as well as the chair of the Department of Microbiology and Immunology. He studied a lot of things in his career. Most recently, he's focused on bacterial pathogenesis with an emphasis on Mycobacterium tuberculosis, the causative agent of the disease tuberculosis, arguably the most important infectious disease in the world. And he has made significant contributions. Some of my favorites are his identification of a really unique way by which this organism tags proteins for degradation and then degrades those proteins, and then exploiting that as a candidate drug target with small molecules, which I think we'll hear about today. Published approximately or somewhere over 300 papers, won a number of awards, just to name a few. He's inducted in the Institute of Medicine, the National Academy of Sciences, and he was the 14th most cited researcher of scientific articles in the 90s, which is amazing. In addition to all these scientific accomplishments, I just wanted to say a quick word about his service. It's clear from his CV that he does a tremendous amount of institutional service. He also does a lot of external service to the community of microbiology and immunology. And most notably, I want to point out his mentorship. He's mentored dozens of postdocs and students who have gone on to tremendous careers and really significant positions. And personally, he and I are both members of the American Asthma Foundation, and they assigned him as my mentor. I don't think he had a choice in that. He got assigned as my mentor. So now I get to benefit from Carl's advice, and I really appreciate you taking the time to do that. So I'd like you all to help me welcome Dr. Nathan, who will be giving us a talk called Bacterial Persisters, Lessons and Challenges from Tuberculosis.
Thank you very much. Our easiest mentoring assignment I've ever had. I've learned so much from him just this morning. And actually, you know, it's pretty standard to begin a talk as a visitor by saying what a great day you've had. But how do I convince you it's really true? I've had an amazingly good day. Really spectacular conversations with people. So I've been interested in host-pathogen relationships for a long time, and that brought me, about 15 years ago, brought me face-to-face with TB, which is a master pathogen that's simultaneously inducing and resisting human immunity. So I thought I could learn something from that about what host immune chemistry is like by trying to understand how TB resists it and so on. And that brought me into a bigger room, the general problem with antimicrobial resistance. So at this point, I look at TB as a lesson book to learn about microbial biology and indirectly about host immunology. But I look at this study also as a potential demonstration project to address some fairly urgent translational problems. So let me give, I know there's a lot of TB expertise and research activity on this campus, and you've probably heard all you ever want to hear about this disease, but I'm going to give you a really quick tour through TB history from a particular point of view, beginning with my vacation in Norway a couple of years ago, and I was struck by how many of the paintings were about people losing family members. This was Edvard Munch, whose mother died when he was 6 and sister died when he was a teenager. This is a picture of that. And it was a very common experience. As you can read, in the 19th century, TB was the leading cause of death in Europe and North America. And there was a lot of despair until Robert Koch discovered the cause in the act of discovering the concept of bacterial pathogenesis along with Pasteur. And this led to a great deal of hope when he found that a culture filtrate of TB injected in infected guinea pigs led to necrosis of the lesions. And the government of Prussia was in the midst of learning about nationalism, and they announced this to the press as a cure, which it was not. But people flocked from all over the world to Germany. There was a tremendous amount of hope. And he went ahead and injected a few people, and some of them died. None of them were cured. He was humiliated. But hope came back when a graduate student named Albert Schatz and his mentor Selman Waxman discovered streptomycin. And this did appear to cure people with TB. And Waxman was so confident he announced that the complete eradication of the disease was in sight. But all those people relapsed with streptomycin-resistant TB, so the cure was not durable. And so monotherapy failed, and then through studies of Dennis Mitchison and others, the British Medical Research Council in the late 50s, 1960s, demonstrated the absolute necessity of combination chemotherapy, and that was introduced through TB to the practice of medicine, where it then entered the cancer sphere, and then, of course, the treatment of HIV/AIDS. And there was hope again. And then the problem of increasing drug resistance, even to combination chemotherapy, led to, this is not a cure, it's not a treatment, it's a control measure, barbed wire. And the point of the barbed wire control measure is that doesn't work either. The patients with multi-drug resistant TB get back to life, family, work by escaping, and we have a really grave problem now. So TB remains the leading cause of death from a curable infection. You shouldn't have to say that in the same sentence. It's a societal problem. And it's becoming increasingly incurable even by combination chemotherapy. So to try to understand that, I think it's useful to go back to why it needs combination chemotherapy in the first place. I think that's actually an entry into some really interesting biology. So what I was taught, and just accepted and taught my students for a while until I had one of those rare moments in the life of an academic when you can think. Obviously it was essential for cure. That was an empirical obligation. And the so-called short course regimen, meaning it took 6 months instead of 2 years, was that you needed at least 4 drugs for the first 2 months and 2 of them for the next 4 months. And this was rationalized based on the frequency of resistance for the drugs. So if the inverse of the product of their frequencies of resistance is greater than the number of colony-forming units or viable bacteria per patient, you can eradicate it, and otherwise you've got a problem. Now, nobody really knows the number of TB in a patient with TB, number of Mycobacterium tuberculosis, that is. It's obviously very heterogeneous because the pathology is very variable. It's becoming a famous problem in the field that we don't really know this. The best evidence I could find in a paper from Canetti in the 1960s was about 10 to the 11th. There's another paper that gives this number, cites a reference. The reference has nothing to do with it, but I'll use this number. The bacillary count in sputum is often 10 to the 3 up to maybe 10 to the 6 per mil. And if you imagine a cavity, maybe 100 mil, I mean, this is a huge number, 10 to the 13. But if you plug in the values for frequency of resistance of isoniazid, mercaptan, ethambutol, and pyrazinamide, which are the standard 4 drugs, that's an enormous computational overkill over the number of bacteria. And if you just stop and think about what that would look like, for time's sake, I won't run through the actual calculation. It's very simple. But it would be, if the whole patient consisted of nothing but Mycobacterium tuberculosis, you would need 6 million of them to equal this number of bacteria. That can't be the answer. It can be part of the answer, but there has to be some other explanation. So a second unusual moment of thinking led to the recognition that this is probably a compartment problem. We have to treat bacteria in multiple compartments, and not every drug is active in every compartment. So what are these compartments? You can imagine they could be quite variable in their nature. They could be anatomic. We could be talking about access to different organs because TB is systemic. It could be different pathologies, whether it's cavities, granulomas, pneumatic areas. It could be on a smaller scale, still anatomic in a sense, but it could be whether the TB is inside another cell or not, which cell it's in, because it can be in many different cells, and even where it is in the cell. If it's in a macrophage, is it in the phagosome? Has that phagosome fused with a lysosome or not, or has it penetrated into the cytosol? But it could be functional as well, the replicative state, or if you will, the metabolic state, and depending on sources of carbon and nitrogen. Or it could be that its growth has been restrained by host immune chemistries or by inadequate access to micro or macronutrients. So in fact, this boils down to trying to treat bacteria in biophysically distinct locations and bacteria in physiologically distinct states. And I think those two things are very closely interrelated, and there's strong evidence for both of them. And I'm not saying this is unimportant, but we now know that these two things are extremely important. So I commend to you Veronique Dartot and her colleagues' paper. It just came out and it's about to appear in the print issue of Nature Medicine, where they used imaging mass spectrometry, very similar to what Eric Skaar does in his lab, on surgically resected lungs from patients with incurable TB who were given all the drugs before surgery. And they find that some of the drugs don't penetrate cavities, and only a few of them do. And in fact, it's correct that there can be monotherapy if given anatomic compartments. And it's not that the combination is present everywhere. So a drug that accumulates in a given site, it has to be effective against TB in the functional state that pertains at that site, and that functional state will be imposed by the conditions that prevail at that site. So in general, the antimicrobials we have today, this is a general statement, are much more effective against replicating bacteria than non-replicating. Occasionally they're effective against both, but in those cases usually much less so against the non-replicating bacteria. In a few cases, it's like metronidazole, it might be flipped toward the non-replicating, but those are usually not very powerful. So the question arises whether that's inevitable. Is that a general inherent property of antimicrobials to be much more effective against replicating organisms, or is it a consequence of the history of how these compounds were sought in screens against replicating organisms? And anyone who's done a screen knows it answers the question you asked, not the one you thought you asked. And if you screen against replicators, you will find what kills replicators. So I'm going to focus on this issue. And the reason I think that's so important begins with this argument that, in general, when people talk about antimicrobial resistance, they're talking about heritable changes. And that's obviously exceptionally important. But that's the tip of an iceberg, and the undersea part of it is conditional, reversible resistance, phenotypic resistance. That's not due to genetic changes, but it's dependent on the ambient conditions. So this was first observed, so penicillin was purified in 1942, and later the same year, a microbiologist named Hobby at Columbia reported that 1 in a million streptococci in an otherwise replicating population resisted penicillin, and she could bring that up to all of them, virtually all, so 10 to the 6-fold increase in resistance by cooling the sample. She didn't comment on 1 in a million, she just focused on the cold cells and said it's obvious that non-replicating strep are temporarily resistant to penicillin. When she warned them, they could be killed in the usual proportion. 2 years later, a man named Bigger in London did the same experiment using staph, got the same result, and he went on to show that he could also increase that proportion of resistance by 6 orders of magnitude by 2 other means besides cold. One was hypotonicity and one was acidification. And again, it was reversible. The bacteria that survived, if then put into log phase growth, were again vastly eliminated by penicillin as before. So because that was described first, I'm going to call it class 1. It's a situation of phenotypic tolerance, that is. It's a situation C where most of the cells are replicated, a small minority are not. For a while it was assumed that the explanation, I'm sorry, I said that wrong, a small minority are persistent. And for a while it was assumed that the explanation was they're not replicated. The first time anyone could actually ask that about a tiny minority was in a paper in Science, I think it was 2003, from Udi Leibler and Natalie Balaban, where they used time-lapse photomicroscopy of bacteria in a microfluidic chamber. E. coli had ampicillin, everybody dies, but not quite. There are a few survivors, run the film backwards. What was their behavior before the ampicillin was added? They weren't replicated. So it seemed to confirm this concept. But then the next time the experiment was done, Science 2013, John McKinney, Niraj Dar, TB this time, the drug is isoniazid. There was no difference in replication rate. The explanation instead was stochastic extinction of the expression of catalase, which is an enzyme that activates the prodrug to its active form. Now, there's a large literature on this. I don't have time to go through it, but basically, sometimes it's associated with non-replication, often it isn't. There seem to be very diverse mechanisms, and to the degree the mechanisms are different, it'll be different bacteria that are resistant to different antibiotics. So what are those mechanisms? I think we could speculate that they could be anything, that where a genetic change gives resistance, but this isn't genetic. It could be epigenetic or some transcriptional or post-transcriptional mechanism. Distinct from that is when almost all the cells, or perhaps all the cells, are not replicating, at least the viable number in the whole population isn't changing over a period of observation. And these cells tolerate almost all the antibiotics we have. And you can do that in a lot of ways. So I didn't put temperature here because in TB it's normal or elevated, it's not low. But you can do it with acidification, with reactive nitrogen intermediates, which are physiologic in the host response to TB, reactive oxygen intermediates, hypoxia, or limitation of carbon, nitrogen, phosphorus, iron, or even sublethal exposure to antibiotics themselves. And this is an entirely speculative explanation. I won't dwell on that, and I'll come with another one near the end. Now, why, when academics stop thinking, they start classifying. So here's an example of, making up categories. Is there really reason to do that? This is what I think is the reason. It's a very practical reason. If different cells tolerate different antibiotics in class 1 phenotypic tolerance, all you have to do is combine different drugs. They have to reach the site, admittedly, but if you deal with that problem. But if the same cells tolerate many antibiotics, you need new kinds of drugs that actually do kill non-replicated cells powerfully, and of course, they also need to reach the same site. So how can you find them? So I didn't know how to think about that. Oh, before I go on, I want to just mention the problem is a little more complicated. And I won't have time to go into that in great detail, but I want to introduce to those of you who haven't already heard the concept. So in class 1 phenotypic tolerance, just to review, it's intrinsic, it's spontaneously manifest, and it's seen under conditions that are permissive for growth. Class 2 phenotypic tolerance is imposed by conditions that impair growth, but it can be subdivided into, let's just call it class 2A, where the bacteria are still viable by a conventional assay introduced by Robert Koch, colony-forming units. Very old assay, 1880. But there are some bacteria that do not grow on aggregate, and yet you can prove they're viable by another means. This can be inoculating an animal, or it can be limiting dilution with back calculation. Rita Colwell, working with cholera and E. coli, in the early 80s, introduced the term viable but non-culturable. But since there are alternative methods of culture, I don't think it's an awkward name, and I'm calling them differentially detectable. And I just don't have time to go into this, but I just want to emphasize its importance because in sputum from untreated TB patients, in most of the patients, these are most effective. They outnumber the conventionally detectable typically by on the order of 100-fold. And we're not monitoring them when we test new drugs by the early bactericidal activity assay. And they're probably not susceptible to TB drugs since non-replicating bacteria resist almost all the TB drugs we have now. Huge problem. We desperately need to understand the biology of these cells better. So anyway, my entry into the optimism that we could find such compounds was accidental. And that's what rescues academics who don't have time to think, is accidents. That's why we should keep working so we can have accidents. So we were looking for inhibitors of an enzyme from intermediary metabolism, the hydrolipoamide transferase. It's part of the pyruvate dehydrogenase complex. And the reason we were looking for that is, I don't have time to go into, but we discovered it's part of the 4-enzyme antioxidant antinitrosated defense. And we thought that compounds that interfere with the defense of the TB against host immunity might help complement conventional therapy. And we found this enzyme is required for TB to be virulent at all in the guinea pig and to resist nitric oxide. And the inhibitor we found, the thioxothiazolidine, actually killed TB only if it was very powerful, whether it was in a macrophage or not. Seemed to sterilize it, but only if it was not replicating. It didn't matter how we made it not replicate. It could be the things I mentioned before. It could be using a cell culture medium that's much more like TB's natural environment than Middleburg-Dubose brought. It could be that TB is in a macrophage that halts its replication. And if we just let TB replicate, it had no effect at all. So that was really very surprising. There are a couple of lessons here. One was thioxothiazolidine is a lousy drug candidate. It's probably going to be promiscuously reactive. It was poorly soluble, had zero bioavailability, terrible PK. We couldn't even do a meaningful treatment experiment. Classic academic exploration of chemical biology from a newcomer. On the other hand, it rang a bell with me that maybe the reason we don't have antibiotics that kill non-replicating bacteria is because we haven't looked for them. And if this compound can do it, how many others could do it? Maybe some of them would be better candidates. So we set out to look. And initially it was quite a challenge to think how to do this, because if your definition of killing is they don't grow, and your condition of the assay is make them not grow, how do you tell if you killed them? But actually, it's not that hard. You need a two-stage screen. So we've been doing lots and lots of cell-based screens under what we hope are physiologic non-replicating conditions. And the Gates Foundation supported this work through a cooperative group called the TB Drug Accelerator. So what we've combined is mild acid, like is known to pertain in the phagolysis of a macrophage, low oxygen, which is true in many lesions in TB, a slow flux of reactive nitrogen intermediates, which activated macrophages can make. The TB can do for itself for physiologic nitrate. When oxygen's eliminated, it makes nitrite and goes on to reactive nitrogen oxide. And very little to eat, and that only a fatty acid, not a carbohydrate. And we score by who grows back when you reverse those conditions and put TB into replicating conditions, who fails to grow back, I should say. And then there's a lot of downstream validation. So the problem here is it's easy to find such compounds. It's very hard to tell how they work. It's very hard to test them without knowing the target. So there's an alternative approach. This is something much more NIH is liking. We don't like screens. And this is hypothesis-driven, and I'll spend the time hereafter on this set of approaches. But the challenge is again getting whole cell activity. So in our first experience with the dihydroaromatic transferase inhibitor, we had whole cell activity. But in many other experiences with target-based screens, one does not, and it's almost bankrupted. It's eroded the interest of the pharmaceutical industry in this approach, and has led to many companies withdrawing from antibiotic development. So just a progress report on the whole cell screen, or so-called phenotypic screen, and this is what we mostly do in terms of person hours in the lab, but it's really boring until it works, and it's very unlikely. By work, I mean you can proof of concept in an animal model, and it's really hard to get to that point. So we've screened well over 1 million compounds, and many of them also in replicating conditions. We've tried to go for chemical diversity. We've used drugs or things that were related to clinical drugs. What pharma calls drug-like collections, which generally are very poorly suited for any bacterial activity. Natural products, when we can get them. Stuart Sharper's diversity-oriented synthesis collection inspired by natural product properties. The molecular library diversity decks that NIH commissioned in the chemical biology programs, fragments, boron-containing compounds, and then these have come from many pharmaceutical partners and from many academic groups. And there are lots of actives. They've devolved into about 6,000 clusters so far. Hit expansion, SAR campaigns underway. There are 2 other screening groups. NIAD, this is Cliff Barry's group. I know he was a visitor here not long ago. an infectious disease research institute in Seattle. And when they get positives in their replicating screens, we look at them in the non-replicating screen. And recently, Merck got galvanized and is doing their own in-house screening and sharing these compounds as well. And we've got 2 of them into mice now. And I don't know if they work or not. And that's the end of this part of the talk, is what is there to say. So let me turn to the target-based screen. And here it's a sort of a chess game that I imagine I'm playing with TB. If I were the bacterium, how many ways could I come up with resistance? And as the experimenter, how many ways can I come up with countering that effort on the part of the bacterium? So I would, if I were the bacterium, I would start with trying to impair host immunity. And the easiest way to do that would be to increase IL-10 to counteract the effect of interferon gamma, which is the key driver, we know this from human genetics, of TB defense, and also TNF. IL-10 suppresses that also, and we also know that from human medicine, a key driver of host defense. So we have a so-called host-directed therapy program in the lab, and we have kinase inhibitors that interfere with the expression of IL-10, and which allow mice to reduce their burden of TB by about a log and a half. It's not great, but this would be an ancillary therapy along with conventional therapy. And in theory, it would be very hard for the pathogen to develop genetic resistance to a host-directed therapy, since the target is not in the pathogen. Unfortunately, the kinase we thought we were targeting, subsequent genetic experiments proved it has nothing to do with it. And so now we have to find the kinase or kinases that are actually involved so we can improve those compounds. So the next thing I would do is try to figure out what the host immune chemistry is, the actual molecules that kill, and break them down. And that's in fact what this enzyme I mentioned before does, together with its partner, lipoamide dehydrogenase, and the 2 other enzymes, HPC and HPD. And as you know, as I told you, we have an inhibitor of that, and it works in vitro, but it's no good as a drug candidate. Next thing you might try to do is adapt to toxic molecules from the host that you can't break down. An example of that is Mycobacterial acid resistance protease, a remarkable transmembrane serine protease. The active site is periplasmate, that without which TB cannot maintain neutrality inside the bacterium when the outer environment is as acidic as it is in the phagosome of the macrophage. Our inhibitors of that screening against that recombinant enzyme led to inhibitors, but they don't selectively inhibit that, so that didn't really work out. But it's a level where you could imagine. After that, you, the bacterium, are likely to have damage to macromolecules, and the next strategy would be to repair them. And we know that from genetic experiments combining host and microbial genetics that the combination of products of the enzymes inducible nitric oxide synthase and phagocyte oxidase mutagenize TB and kill it in a mouse. And TB survives to go on to eventually kill the mouse only if it has nucleotide excision repair. So we developed, I think, the world's first inhibitor of nucleotide excision repair in vitro, target-based screen, doesn't enter the bacteria, useless, but it's another level of concept. What's after that? Well, now you might have irreparable damage to macromolecules, and generally that's associated with loss of the normal function and often toxic gain of the new function. For example, irreversibly oxidized proteins unfold and expose hydrophobic areas and bind illicitly to other targets, so you want to degrade that. And TB has a proteasome that contributes to that. And then we imagine that after that there might be one more strategy, or 2 more actually. The next one might be to sequester what you can't degrade, wait for bitter days. When division begins again, keep all the junk in one progeny cell, let the other one be born clean. And this would be a form of aging and rejuvenation, if you will, or senescence and new birth in a single-cell organism that divides by binary fission, which we think of as being indefinitely immortal. heritable and be a different way of thinking about it. This actually occurs, and so I'll talk about these last two in just a second. Recently, we've been adding one more layer to this, which is TB might then give up on all those other remedies and drive itself into a deep, non-replicating state where you don't get colony formation. And again, wait for another day. We know almost nothing about the biology of this. So let me focus on this story. As they're foretold, I do want to tell this story, at least briefly, even though it goes back quite a bit. So a genetic screen carried out by Erin Darwin, who's now a professor at NYU, when she was a postdoc in my lab, led to the identification of candidate genes for proteasome in TB as being required to recover from a nitric oxide-based stress. And this was pretty upsetting because this isn't an enzyme you think of as like, thyroidoxin system and methionine sulfoxide reductase system as having an anti-nitrosative or antioxidant function. And also because bacteria were not known to have proteasomes, so there's a significant challenge to this concept. As part of that workup, we asked Millennium, as it was called at the time, not Takeda, for Velcade, also called bortezomib. This is a mammalian proteasome inhibitor, a peptidylboronate that is used it was first approved for the treatment of multiple myeloma and then certain lymphomas. It's very toxic, but very helpful to those patients. And they said no. I guess they were afraid we might learn something that would threaten the market value. But they were very generous in giving us a conjugator where the pyridine is replaced by a morpholine with a phenyl by a methyl group, and also it's an antimer. So this compound inhibits human proteasomes, this one doesn't. This compound inhibits a proteolytic activity in TB lysate and prevents TB from recovering from the nitrosative stress, and this compound doesn't. So that encouraged us, if we could validate the target genetically, since it wasn't even known to, these genes weren't even known to encode the proteasome. That's one. Number two, if we could find compounds that were species-selective proteasome inhibitors, there's no way you're going to give this toxic anti-cancer drug to a patient with a curable infectious disease. Maybe we have some room to move. So Gong Lin, who's a chemist, by the way, learned some biology and managed to express, or to get E. coli, I should say, to express this 28-mer protein as a product of two genes, proteasome component B and A. And this is what it did to itself. It turned into this particle that William Lee and his colleagues at Brookhaven solved by cryo-EM and also by crystallography, as I'll show you. And it looks like a mammalian proteasome, except the entry door is closed. And we speculated that's because there's companion proteins that open it as a regulatory step, and that story is now well defined. I don't have time to go through it. They exist. And so he also made a deletion mutant of the octopeptide from the immunoterminus of the alpha chain to get the so-called open gate port with which to do a screen. The crystal structure showed that the mammalian, Velcade or bortezomib, the mammalian proteasome inhibitor actually alkylated through the boron group. I'm not sure that's the right term. The hydroxyl of the N-terminal 3. The reason you can't actually see that here because it's not close up enough, although you can actually see part of the molecule hanging out. But the reason that's important is to introduce this class of proteases, which is very uncommon, N-terminal 3 hydrolase. So the active site residue is this very last first hydroxyl in the amino-terminus of the beta chain. So here's a genetic validation. It was carried out by a student Shilgan Lutra in the lab of my neighbors, colleagues, Dirk Schnappenberger and Sabina Eric. This is a control. This is the classic log expression of CFU versus time sort of hockey stick pattern you see in an immunocompetent mouse when you infect with low-dose aerosol infection. The TB replicates logarithmically until about 3 weeks. At that point, the transcript level in the lung of inducible nitric oxide synthase is 1,000-fold increased. There's a similar increase in interferon gamma, TNF, interleukin 12. So host immunity sets in and you get this non-replicating state. There's a little fluctuation, but it goes on for about a year while pathology slowly mounts until the terminal event. So in this infecting strain called H37RB. We've installed the reverse TET repressor but no operator, so this is a control. And they fed the mice doxycycline, and it doesn't make any difference, so the doxy has no microbial effect. But when they put the TET operator upstream of the proteasome operon beta alpha chain and fed doxycycline, there's about a log loss of CFU per month, which is about as good as the best drug gives by itself. So this would be a genetic target validation criterion. You can check that box. So Gongwen did another target-based screen. Again, we're told not to waste our time. Occasionally it works. And he found an unusual nucleus, chemophore oxofluorescent. The chemist will immediately recognize liability. Nonetheless, there was an SAR, so this one worked, this one worked, this one didn't work. At equimolar concentrations, they're better on TB than Velcade or bortezomib. Not terribly potent, but within the range of classic antimicrobials, and very rapidly entering and acting. And they had the property, bortezomib weakly, and these other compounds much better, the property of killing TB when it wasn't replicated. In this particular experiment, it's a nitrosated stress, no impact on replicating TB, and it was probably obvious from the previous slide, I didn't mention it, knocking the genome or suppressing it as they did in that experiment. Either way, there's very little growth phenotype under normal conditions. So it's not an essential target. Classically, it would be passed over in an antibacterial program in industry because it's not essential. But my whole point is essentiality is conditional. Depends on the condition. So here's Velcade killing eukaryotic kidney cells, and these compounds are vastly less toxic. And the reason is because they don't inhibit mammalian proteasomes. So how do they work? So this experiment, with the help of Eichen, Denning, and Rockefeller, peptide mapped the proteasome before and after treatment with either of two compounds. I'll show you this one. And there was no change in any peptide except the immunoterminal heptapeptide of the beta chain. So lots of things were exactly the same. So what they actually saw was a mass shift of 25.98 Daltons. Now, it was Heitem's idea to try reductive glutaraldehydation. That's a word, I think that was the first time I said it successfully. And that will react with primary means. And this particular peptide grew by 68 if the whole proteasome had first been exposed to this compound. But it grew by twice that if it had not first been exposed to this compound. So you put all that information together. There's two primary amine opportunities, the N-terminal amino group and the lysine epsilon amino group. And we know the activity dies after this treatment. We know the activity is dependent on, on this hydroxyl. And the only way we could fit all this together was cyclocarbonylation between the hydroxyl, between these two residues, atoms. So Weill and Lee then confirmed that that was true. This oxazole forms, that's rock stable. And that is going to kill the proteasome. But it leaves the mystery of why there's species selectivity, because we were warned we couldn't find species selectivity because the active sites are very similar, the mechanisms are very similar. And how did we lock into this? So that depends on the chemistry. And so this chemophore, the carbonyl, is attacked by the hydroxyl, whose nucleophilicity is enhanced by this visceral amino group, and you get a tetrahedral intermediate that can break down on either side of that oxygen-carbon bond. And then everything depends on what happens next. So the easiest thing is for water to come in, whichever way this broke down, and regenerate the threonine intact on the proteasome and spit out the dead inhibitor. So in that case, the inhibitor is just a substrate, and the enzyme is turned over and it's ready for action. But if the amino group gets there first, that is the threonine amino, and completes the cyclocarbonylation, the inhibitor is gone too, so it's a suicide inhibitor, but the enzyme is permanently dead. The only way to fix that is make a new copy. So why would that route be favored in TB and not in human? And we think the answer is somewhat speculative, but it's coupled to a striking structural biology observation that Weill made, there are many, many proteasome structures in the literature, but no one had ever seen before a shift of this switch 4-helix 1 loop. And there's a very substantial drop over the active site in the case of the TB proteasome. And it turns out the hydrogen bonds that mediate the position of the loop before it shifts and the ones that mediate the position of the loop after it shifts are all not conserved between TB and human. So we think, it's very hard to carry this argument further, but we think that's the explanation for why the active site is, this shift is enough to keep water out of the active site, I should have mentioned. So we think it's keeping the powder dry, if you want to use that metaphor, and the residues are distant from the active site that control its specificity. Recently, another group tried another way of making TB not replicate. This is just carbon-nitrogen starvation, and they were able to eradicate it with another version of oxythiazole. So I think the lessons from this are as follows, that against the precept that you should only target proteins that the host doesn't have an analog of, and that precept is widely ignored with conventional antibacterials anyway. Here we were able to achieve 1,400-fold selectivity versus human proteasome. I'm just mentioning the subunit that's targeted most importantly by ortezomib. It's the beta-5. And this arose from very subtle differences which were not in the active site, which were at a distance. And this is the second example after the DLAP inhibitor of a compound that only kills a bacterium when it's not replicating and does so in diverse non-replicating states. So more encouragement that such compounds are possible. This nucleus is also not a drug candidate because of its instability, but again, it's an encouragement. And I think perhaps more importantly, it's the first evidence of a whole other way to kill bacteria. So existing antibiotics work in very limited numbers of ways, inhibiting the synthesis of nucleic acids, proteins, cell walls, folate, or very recently some membrane perturbing agents. And here's another way, inhibiting protein breakdown. And that opens up thoughts of doing this other ways. So what about better candidates? So Millennium went on in their anti-cancer program to introduce these so-called NC-cap dipeptides that are remarkably selective among classes of proteases for the eukaryotic proteasomes. And Gong Lin, actually they're very generous, they invited him up there to screen their collection of these compounds against recombinant TB proteasome. And they gave him the ones that didn't work in their mind, that they had no interest in, among which were several that did work on TB. And we could not reach a global access-based intellectual property agreement with them, so we took none of those compounds. Gong came home and made different ones. So this is dipeptide NGON2, which is 50 nanomolar for TB, and that selectivity is almost 5,000-fold. And he also looked at the immunoproteasome, the new subunit that comes in, that's already present in the immune system and comes in other cells during immune responses. High selectivity there as well. So why is this one selected? So Weill and Lee has just determined the crystal structure of 6 different versions of this, and we think the answer again is subtle, but this time it's not in the active site proper, but very near to it, S1 pocket and S3 pocket. And in the mouse, this is just modeled, of course, it's not directly observed. In TB, it's as observed. And it's just differences in the shapes of the pockets. I won't go through the atomic details. They're enough. So this pendant diethylamine clashes, or is predicted to clash in the eukaryotic and fits snugly in the TB. And the same with this bulky group, which is very important for the selectivity of TB, not room for it in mammalian. So the lessons we've got from this continue, and there are surprises for us. So in terms of prospects, we've now gotten the attention of global lines for TB drug development that maybe there's hope. And they've brought a contractor, Schrodinger, to do sophisticated molecular modeling. And through the Tri-Institutional Therapeutics Discovery Institute that Weill Cornell shares with Rockefeller and Sloan Kettering, we've got access to a company called Cyclofluidic that has a very rapid iterative program for combinatorial synthesis and inline assay. and they went in a couple of weeks, a couple of orders of magnitude improvement in potency. So we have single-digit nanomolar thousands-fold species selectivity, and we've gone away from the peptide backbone. Another accident, so to speak, along the way, we discovered killing Plasmodium falciparum with extreme separation from human cell activity. This is a well-validated target. There are many papers in the literature, including treating mice with rodent malaria. But all those compounds have human proteasome activity. They're all toxic. None of them are candidates. So we're actually very excited that this might bring malaria proteasome into the realm of reality as a target. And then accidentally discovered while he was looking at selectivity some examples of the series, but they're different individual compounds that are nanomolar inhibitors of the human immunoproteasome, but they don't inhibit the human constitutive proteasome. Some of these prolong cardiac allograft acceptance indefinitely in mice. Some of them, the same one actually seems to treat a model of inflammatory bowel disease. So this has nothing to do with TB, but it's another example of what happens to academics when you stumble into something new. So in the last couple of minutes, I want to talk about this topic, what's next if you can't degrade a protein because it's too badly oxidized, for example, or nitrosated. And this is work of Julian Malbergex, a postdoc in the lab. And he's the one who demonstrated that it is possible to sequester and then asymmetrically distribute upon resumption of replication these, what we call IOPs, irreversibly oxidized proteins. So just to summarize what he found, and then I'll just give you a touch of the evidence in the interest of time. TB in stationary phase, or exposed to an oxidant, or given treatment at sublethal levels with an antibiotic that inhibits function of the ribosome. This is an aminoglycoside, kanamycin, well known to produce malformed proteins. Increases the abundance of IOP and Mycobacterium. This can be measured roughly by accumulation of carbonyls, but to the degree that they're irreversibly oxidized, they become irreversibly insoluble. And so these are not quantitative measurements. What he's seen at the single cell level is these form small aggregates. And those aggregates coalesce to form a smaller number of larger aggregates, which increases the chance that they'll be asymmetric. I mean, if you have one, it has to be asymmetrically distributed. But even when it's a number greater than one, you find this asymmetric distribution. So he can trace these with a fluorescent probe for carbonyls by identifying individual proteins to which this happens and then installing a fluorescent tag fusion protein in the bacteria. Or it turned out that there's a chaperone. This is the equivalent of one of the HSP70s in mammals. It's called ClpB. And by putting in a fluorescent version of ClpB, it turns out ClpB joins the aggregates. It's not itself a target of irreversible oxidation. It's not covalently attached to the aggregates. It's actually comes in and out of the aggregates. He was able to show that with fluorescence photobleaching experiments. And its ability to join the aggregate depends on its ATPase activity because point mutants abolish that. So this is actually the easiest way to follow the aggregates. So when stress is relieved, the aggregates, sometimes in some bacteria they resolve. In other ones they distribute, as I said, asymmetrically. And that has a huge impact on the fate of the cell that inherits them. So in proportion to how high the load is of IOP, that cell is going to not grow again, or it's going to explode, which we take as morphologic evidence of death. And it can do that spontaneously, or is very likely to do that to the next pulse of this otherwise sublethal dose of antibiotic, imitating the way drugs reach bacteria in the patient who takes the pill once a day. In contrast, the progeny that are born without this burden survive very well and grow the fastest. So this whole process could be a drug target. If we could somehow block the chaperone or block the asymmetric distribution, if we knew more about it, this might reduce TB's ability to survive antibiotics during non-replicating states, which is to say, might block the display of class 2 phenotypic tolerance. So this, I'm going to go really fast through this now, because it is published recently. This was the hydrazide probe that Gong design. He wasn't able to make it well enough, but Wojtek Orfelli and Gwen Lee Young at Sloan Kettering were able to separate the product from reactants enough to make a very clean version of this. And you can put your fluorochrome of choice on this. And, sorry, I went the wrong way. And this is what he saw. So let's start in log phase. The tagged chaperone clip B is uniformly distributed. The probe for fluorescent proteins, you can't tell from this, but I'll just explain it sticks to the outside of the cell. And that if you do confocal sections, the inside is clear, so this is the worst that it looks, and there's no overlap. In stationary phase, the clip B forms aggregates. Now looking through the midsection of the cell, the carbonyl proteins, carbonyls I should say, seem to form aggregates, and these coincide to a significant extent. So if you look in a time lapse, I'm not going to show you the movie, but just some stills. This is before treatment. It looks like the clip B is already aggregated, but it's sort of patchy. After sublethal kanamycin, this is what the aggregates look like. And actually the amount of clip B is exactly the same because Julian was is judged by pixel counting fluorescence intensity. There's no difference. It's just the standard deviation of distribution over space skyrockets. It becomes very heterogeneous. So this is early on. And then as I say, these aggregates fuse. They tend to move to the pole. And here's an example of a single aggregate at the pole. The cell divides. One doesn't have any, one has some. This one pops, this one's fine. We'll go on to repopulate the culture. So at the whole population level, there's a phenotype to knocking out clip B. So again, if you look in log phase, so this is the wild type, the knockout, the complemented strain. There is no phenotype. Classic growth curve, not an essential target, not of interest in the conventional approach. But if you look at late stationary phase, the wild type and complemented have a very brief lag. But the knockout takes 3 weeks before you can see any growth at all. We actually thought for a long time it was completely dead, but fortunately Julian waited a little longer. Some cells survive. So many of them die, some survive and repopulate. And if you look at the fluorescent probe for carbonyls, it just rockets up in late stationary phase for the clip B knockout. So the clip B isn't just a marker. It's functionally relevant for handling these proteins. And in mice, there's about a 2-log deficit after a few months in the bacteria that lack this chaperone, and they have much less pathology, as you'd expect. So now we go back to a more up-to-date version of what's going on for phenotypic tolerance. It's very popular right now to talk about this being controlled by toxins and antitoxin pairs. And one way of unleashing a toxin is when there's proteolytic degradation of its cognate antitoxin. So this has been shown to happen to account in some cases for class 1 phenotypic tolerance. And there can be altered gene expression, and this can include upregulation of pumps. It's not necessarily growth arrest, it could be growth arrest. And I'm just going to put forward the possibility that this might be the same thing that's going on in class 2, but just more of the cells, and associated in this case with growth arrest. So TB has at least 70 toxin-antitoxin pairs, probably 90, but it's hard to be sure about all. Messiniv, who's one of the people who discovered this, I'm sorry, Ken Gertis, working with Messiniv. Gertis was one who discovered this process, one of the people. Has classified them into type 1 and type 2. Type 1, the antitoxin is an RNA transcript. Type 2, which is more familiar, it's a protein. And the way type 2 works involves a degradation. So in E. coli, this is by a chamber protease that TB doesn't have, called LON. And the activity of LON is controlled by polyphosphate, whose synthesis, the balance of synthesis and degradation, is controlled by REL-A in response to a gene that's transcribed in response to stress. So TB doesn't have LON, but it has 2 chamber proteases, the one I've been talking about, the proteasome, and another one called CLIP, caseinolytic protease, is the origin of that CLIP P1, P2, again, 2 rings, 2 heptameric rings. So just taking that all into account, going back to my 7 ways I can think about, if I try to imagine on TB, how can I resist the host? 4 of these could be involving proteostasis or proteolysis. Here, instead of DNA, we could just substitute a protein that needs to be repaired. And so this has led to our current enthusiasm, which is to try to hit this pathway at multiple points. So we're trying to target, hasn't been time to introduce all these players, but DNAK, which is another HSP70 conjugate, ClipE, the proteasome component, ClipC, which is the controller for what gets into ClipP1, P2, and ClipP1, P2 itself. So an activator of ClipC, natural products like ecumycin and cyclomerin have been found, and they enter and kill. That's work of Scott Franzblau and Kim Lewis and Fred Goldberg at Harvard. Goldberg, Eric Rubin, and Bill Choch, and so Harvard, the Tufts people, have found peptidyl borinates that hit ClipP1, P2, and we've shown that don't hit the TB proteasome. We don't yet have inhibitors of ClipE or DNAK, but we're working with Tuberculosis and Sloan Kettering to take the ones that are cognate to her human HSP70 inhibitors that don't hit human and look at those. And conceivably, this could also prevent entry into this deep, non-replicative state called the differentially detectable state. So I just want to close by saying TB is endlessly fascinating to me, and the biology seems to be infinite, even though there's only 4,000 genes. But it is the point of the spear, if you will, of a bigger problem, of the general problem of antibiotic resistance that's confronting all of us, no matter what well, there's at least a dozen infections, some cases of which are now untreatable by anything. A dozen species of pathogen. Numerically, the largest example is probably TB, but gonorrhea was one of the first. I'll go a little farther. It's the first infection you could cure with a single shot of penicillin. And now there are cases you can't cure with anything. And the consequences of that can be infertility, ongoing pain, arthritis, sepsis, and death. Yeah, it's a big problem. And so the point I want to make is that the problems are general, and they involve, in every case, both heritably and phenotypically drug-resistant bacteria. And there's also an organizational problem for us as scientists or physicians, and it's how to work in a cooperative way to do this. We can't do this without drug companies, but they've mostly given up on doing it. And I think it's very important to think of cooperative approaches. Just came out this look back at 2 examples where everybody got together and solved the problem. They both occurred during World War II. One was the penicillin project, the other was the malaria project. And now I think the TB drug accelerator is a new example of that. When it was created, no one remembered these old examples. And there's another example discussed here called the Trace Cantos Open Lab Foundation. So I'll stop there, and I just want to thank collaborators, and there's so many, I won't take time to go through everybody's name, but I focused on the work of Gong Lin and Julian Valverdex. And I want to thank the Gates Foundation for funding the Milstein program, which is local to Cornell, and NIH for what's now called the TB Research Unit, which is taking over from the TB Drug Accelerator. Thank you.